Okay, I thought maybe kind of just doing a real quick review over mitosis and meiosis on the PowerPoint may be a little bit helpful for some of you. Meiosis seems to be a little bit confusing for people. I can always tell by reading some of your when you post some of your discussion question answers. Although I would have to say that you know you're supposed to post two responses to your classmates' post, and I love it how you post responses to your own post. Like, I was so great, I was so good at this, I was so impressed with myself. Man, am I good or what? You know, I read those first actually, and my friends, I have them read those when they're stoned because they think it's really funny. Those are really good things to do. Um, the friends do that because those are great. And many of your answers are really good anyway. But you know, when you take them right out of wherever you're getting them and do a copy paste, I don't really know that you're getting a whole lot out of that. We'll find out when you take the test. You know, unless you just memorize that stuff, because I know it came out of some book and you're just copying and pasting. Anybody can copy paste. That's not really what we're trying to accomplish here. But if you find that valuable, copy paste activity. Okay. I'm more into the understanding the concept kind of a thing myself, but that's just me. Okay, so yeah, you're supposed to post to like other people's posts, not your own. But those are funny, and you've been doing them all semester, so I guess it's a little bit too late to complain about it at this point. I've just been laughing so hard that that I really didn't want to tell you about it. So semester's almost over, so it's totally cool. Not right, but pretty funny. So as long as it's funny, I'm pretty good with it, really. I, you can get away with a whole lot of stuff as long as it's funny. Okay, so we need to be able to reproduce, obviously. So we do cell divisions. We use chromosomes, carry genetic information, and we get really what we call homologous chromosomes. You get one from your mother, one from your daddy. That's pair number one. One from your mom, one from your dad. That's pair number 2 and 3 and 4 and 5, so forth. We actually get 46 pair of chromosomes. And there are two ways to reproduce asexually. Now, we don't really reproduce asexually, other than individual cells can reproduce asexually if you want to take one cell and divide it into two cells, which is what happens if you damage yourself, if you cut yourself, for example, fall down, scrape your knee. You need to replace those cells. And of course, you need to replace them with the same cell type that was destroyed. So that is an asexual type of a production, basically, because you take the one cell, or the cells on the perimeter that are not destroyed begin to divide to create the cells to replace the damaged ones. So, you know, that's done all the time. But of course, if you want to reproduce and have another organism, you know, as animals, you can't, you know, you just can't spit on the floor and that would actually develop into another organism. It's not going to happen. Although when you spit on the floor, you're going to have some of your oral cells, which will have the 46 chromosomes. So yeah, genetically, all of your information is sitting right there on the floor in that saliva. It would be there, but it isn't going to develop at all. So anyway, asexual reproduction happens a lot. We use this a lot in plant propagation. So if you want to take a plant and produce two plants that were exactly the same, you can take cuttings. People do that. Lots of plants do that automatically. They create bulbs. Like if you plant irises, they grow up from a bulb. Or I like to plant what's called society garlic. I don't know if you've ever seen it. It's kind of a native plant, but and I grow mostly natives. But it has a little bit of a garlic odor to it, but not much. You'd have to get pretty close to it. But it produces a really nice looking plant, and then they produce really nice, beautiful purple blooms on them. But they're also by bulb, and so you can go in there and like break them up, and then of course those continue to divide and create more society garlic. So I tend to have quite a bit of society garlic, but and it works well outside too, actually. I do a lot of it indoors. Just what I do. Anyway, different ways to sexually reproduce, asexually reproduce. A lot of organisms, like if they're one-celled organisms, that's how they reproduce mostly. If you have an amoeba, it wants to become two amoebae. It just simply divides, absorbs enough nutrient so that it will divide to become two amoebae at that point. So, yeah, asexual reproduction. And we talk about binary fission when we get into the bacteria. So sexual reproduction, anytime you're doing a sexual reproduction, you're creating a genetic variant, something that is a variation of the mother and the father, and not really just 50% mom and 50% dad, but very closely that. Very rarely would it be exactly that, but it would be very, very close to that. And, of course, that is to create genetic variability. And that is what really gives us the ability to evolve, is to have that genetic variability built into the population. So without that, we would not be able to evolve. So it's very important. And so whoever came up with that beautiful idea called sexual reproduction hit on a good thing. Very, very smart. And the way that works is really beautiful as far as I'm concerned. So cell division, if we're talking asexual reproduction of one-celled organisms, and then of course the growth of us from one cell, the fertilized egg, which would be the zygote, eventually we divided to where we are today. Replacement of dead cells, repair, and then of course the production of sperm and egg, that is a meiotic cell division. So really that's just not even that's not asexual at all in that case. Okay, what you're looking at here, this is a yeast, and the yeast goes through mitosis, and really what it does is create a little bud, and that bud will eventually pinch away from the parent cell, but it has the exact same number of chromosomes. It's a clone. They're exactly identical. Some other ways that organisms asexually reproduce, you may not be familiar with the regenerative powers of a sea star. A lot of people call them starfish, but certainly they're not fish. But if you were to, say, chop this sea star here into, say, all of these different finger-like looking leg projections, as long as you retain a section of this central disc area, then each one of these legs will regenerate in a completely new sea star. They do that. All you got to have is a little piece of that little middle section there in order for it to do that. Sea stars feed on clams, and so the clamming industry at one point thought, oh, this is not good. They're eating all of our clams. So what they would do is they would dredge them up, the sea stars, and they'd cut them into little pieces and throw them back out in the ocean. They just created more of a problem. They didn't help themselves whatsoever. So once they found out, oh, we've got more sea stars now, and they were pulling them up, in pieces like this one here that basically is a well-developed leg and then some other small legs coming out of that. Didn't take them long to figure out what was going on. They were like, wow, man, that thing can regenerate like crazy. Be like, us losing an arm and regenerating a new one, although that's coming. It's coming. I've seen them grow a human heart in a laboratory. A human heart, a real one, functioning human heart. Crazy, yeah. We're getting pretty near the capacity to do such crazy things. Here you have a leaf that was stuck in garden soil here, and it has put on roots, and then from those we've sprouted new leaves. That's an asexual propagation. Here you have a whole bunch of sexually reproducing individuals, although maybe he shouldn't be involved, or her. If that is indeed a he or a she, I'm not making those judgments at all. Don't want to throw that on them, give them any preconceived ideas as to what they're supposed to be. I'm funny. All right. Okay, I know. There's only a few you could think I am, but it's good. I'll take just a few. A few is good for me. I'm all over that. All right, this is just a cell dividing into two cells. Okay, so binary fission, this is what your bacteria do, and that's what they typically do. Now, they can sexually reproduce, not all of them, but some can actually sexually reproduce, meaning they can exchange genetic material. Obviously, they have to join, and through the pilus, if you remember that, that's way back when, but anyway, the pilus, they can exchange genetic material and then have a recombination. That can happen. But most of the time, bacterium just kind of lays around, soaks up nutrients. They release digestive enzymes that are released out of the cell and then digest whatever they're on, and then they absorb that, and then they get bigger and longer, whatever, and to a point to where they just simply divide into two units. Very simple mechanism. Prokaryotic cell division. At one point, you do have to duplicate the chromosome, and then the cell has to elongate to pull the two chromosomes apart, and then you divide the cell. Now, if you want to make it sound more complicated than that, you're welcome to, but it's really not necessary. Not necessary for me. You know, if you get a discussion question like that, yeah, that's good. Not a complicated event at all. Okay, now our cell division, which is basically the asexual, the mitotic cell division, which really is now referred to as the cell cycle, because really you've got one cell that divides into two, and then they have to grow, and then they divide, and you get 4 cells, and they divide, then you get 8, and then eventually you're 70 trillion cells. Somewhere along the line, they begin to differentiate into eyeball cells and, you know, nose cells and lip cells and tongue cells, and we could go further down if you'd like. We could make our way, you know, piece by piece through the body. Yeah, we could. Anyway, so one thing that is certainly going to complicate the situation is you have a nuclear envelope, a double membrane. You're going to have to get rid of that thing, because you're going to have to replicate your chromosomes, so they'll all look like X's, and they're called sister chromatids, as we understand, and one will go to one side of the cell and one will be pulled to the other side of the cell. So they have to replicate, and then you couldn't pull the sister chromatids apart or pull them to the poles, and in reality they're called sister chromatids as long as they're attached. As soon as they detach, each is referred to as a daughter chromosome. But anyway, so the daughter chromosomes are what actually go to the poles, but you have to get rid of the nuclear envelope to be able to do that. So that's what we have to do here. Initially, in an interphase, all the chromosomes are a non-distinct mass. You can't really distinguish a chromosome from another chromosome. They're not coiled to the point that they're visible, and we call all that chromatin. So a lot of this is just review for you guys. But anyway, now at this point they have condensed. I can see them. So I know I'm in a prophase at this point. And that's probably the nuclear envelope surrounding those. So we haven't actually eliminated that nuclear envelope where we can pull the chromosomes to the poles just yet, or begin to line up the chromosomes through the center of the cell. So here they show you the 2 sister chromatids. So we have 1 chromosome, and of course we actually have 46, but they're just showing us 1. And then it's going to replicate, so we have 2 sister chromatids held together at what they call a centromere, and then they are pulled apart. The spindle fibers, which are microtubules, attach and then get shorter and shorter and shorter, and slowly they pull them to the poles. But of course, they are sister chromatids as long as they're attached together, and soon as you pull them apart, these are daughter chromosomes. Now it says separation of sister chromatids, but it didn't say into daughter chromosomes. I wish they'd put that. Separation of sister chromatids into daughter chromosomes. That would have helped us. They just said they go into daughter cells. So for most people, they still think these are sister chromatids. They're not. Technically, they are not. They're daughter chromosomes. Just make that point. Okay, so we do the cell cycle again. We've already demonstrated that. So you have to go into interphase. That's where you get the growth. So this immediately happens after one cell divides. Now, do they continue to divide over and over and over and over? No, they don't. I mean, you know, your nose will eventually stop dividing. Your toes will eventually stop dividing. We could swing from trees. That could be interesting. I would love that, actually. or a long tail, I could swing from trees. Up there with the monkeys. It would be super. I could have a lot of fun. Anyway, God saw fit that I would be here to torture y'all with this. I know. It's sad, isn't it? Okay, so interphase anyway. So one cell divides into two, and then each one of those will grow to the original size. Well, the period during which they're doing that growth is called a G1. And then we have to replicate those, meaning that they're all going to have to, all the chromosomes are going to have to form sister chromatids. So that's the S phase. Duplication, G2. That's typically where you make a whole bunch of proteins and enzymes, which are proteins, in order to divide the cell. Now, mitosis, which is the mitotic phase, the mitosis is really the division of the nucleus, meaning you're carrying one set of chromosomes to one side of the cell and one set to the other side of the cell. And the mitotic phase also includes cytokinesis, which is really when you divide the cytoplasm, which is what cytokinesis means, cytoplasmic division. Now, if you're asked to diagram mitosis and you give me one of these on the discussion question exam, you will get 0 points. This is not the diagram we're looking for. Oh, you're right. You're right. You won't even have a discussion question for Module 4. That's right. I forgot about that. We don't even have time for it. That's right. Anyway, so we don't have to worry about that. Sometimes I get those. People will like do a diagram, and all of a sudden it's like, I'm going, you're really going to give me this a diagram for mitosis? Anyway, these are the phases. Prophase, now use prometaphase, metaphase, anaphase, telophase, and then cytokinesis. All right, so the video won't work, but we can go to here. Okay, so interphase, your chromosomes are not distinct. They're in a loose, uncoiled state. If you're an animal cell, you do have centrosomes, which is the area around the centrioles, which is where the microtubules form in the centriole. The centrosome area. And then they begin to migrate, and then by the time you get into prophase, the chromosomes have condensed. Well, they already look like X's here, so when did they become X's? They become X's in the S phase of interphase. That's when that happens, S phase. And then you produce all the proteins in in the G2 phase. And then you get into prophase. So in the prophase, I don't know where that came from. In the prophase, then that is where you condense your chromosomes, and they are visible. Now, in prophase of mitosis, you don't pair homologous chromosomes. That does not happen. So it's just a random arrangement of chromosomes. And then by the time you're getting into prometaphase, you've lost your nuclear envelope, or you're losing it. It just uncoils and goes into the cytoplasm. And at this point, the chromosomes are going to attach. They're at the centromere area to something which they call a chromatochore. A kinetochore. Look at that, kinetochore. That's important because what happens is every time a cell divides, you divide one cell into two, and then that cell divides again, and it divides again, and it divides again, and it divides again. Every time you're dividing that, the kinetochore gets smaller and smaller and smaller and smaller. At some point, it won't divide again because the kinetochore is too small. That's called aging. That is what causes the aging process. So cells stop dividing. So your skin gets thinner. You've got all these pains you didn't have because you can't replace the connective tissue, the tendons where your muscles attach to your bone as rapidly because their cells aren't dividing. They don't divide. You don't have as many dividing. So you end up with pools of tendons, which that hurts, and it takes a long time for those to heal. Very easy to snap an ankle or break a hip, or They end up with all kinds of problems. And that's it right there. The kinetochores, every time the cell divides, the kinetochores get smaller and smaller and smaller and smaller, and then they don't divide anymore. So you start losing cells is really what happens. And of course, we understand that's aging. Most people don't know what it is or how that happens, but that's really how it happens. Pretty simple. They are developing drugs now that will keep the kinetochore from getting smaller. If they can do that, guess what? They expect the lifetime of your children to be maybe 1,000 years. I mean, in good condition. Well, no, if you eat them, then at least hopefully you barbecue them where they're tasty. Yeah. Just pull the meat out and barbecue it. Fillet of 14-year-old. Because it will be at the age of 14 that you will want to fillet them. Yeah, just so you get that from me. Yeah. You go ahead and write that down. Fillet child at 14. It's a joke. I have to always postscript because you never know. Somebody in here could be an absolute nut and go, he said we could fillet our children. Take me to court or something, Those people are out there, I'm telling you. They're roaming around. They're amongst us, hopefully not in this room. Y'all have heard me throw out enough crap out of my mouth that I think y'all understand when it's crap and when it isn't. But I have run across people who did not know the difference between a joke and just reality. Like, you're living in the non-real world. What's that like for you? Okay, so prophase. Chromosomes condense, become visible. Basically, that's what happened. Prometaphase, nuclear envelope breaks up, disappears. Your microtubules are going to begin to form, and they connect to the kinetochores. As long as they're not too small. If they're too small, they can't connect. Therefore, the cell can't divide. Therefore, you're aging. All right. So, and this is what you're showing here. Then the microtubules line up the chromosomes through the center of the cell, and then the microtubules begin to shorten. They used to think that they shorten at the poles. They don't now. They think, in fact, they're pretty sure they shorten right there near the chromosome. There's an enzyme that breaks down the microtubules, and therefore it actually gets closer to the poles, is how that works. Then when you get into telophase, you're reforming your nuclear envelope. The chromosomes will begin to decondense back into chromatin, and you're having cytokinesis take place. So you're basically done at that point. And you have all this. I'm not going to read all that to you. So cytokinesis is different whether you're dealing with an animal or a plant. Animal, you do pinch the cell into two units. There are microfilaments. You may remember the actin molecules. That's really what that is. They're actin. And they begin to pinch into, smaller and smaller units until eventually you pinch the cell into two units. You have your two daughter cells. So you create this little furrow here called a cleavage furrow, sometimes called an animal furrow. Plants do something completely different. That animation won't work. Okay, so if you're dealing with a plant, what it does, since it can't pinch into two units because of the rigid cell wall, you have Golgi apparatus will begin to form these little vesicles that line up through the center of the cell, and that's called a cell plate, not a cell wall. The cell wall surrounds the cell. This is a cell plate. And the cell plate continues to develop, and of course, until you have formed two daughter cells. Okay, then they talk a little bit about cancer, one of our more favorite, lovely subjects. 20% of us will actually die from cancer. That's 1 in 5. Look around the room, 1 in 5. I will probably be the first to go out of this group. I smoked cigarettes for a long time, and I've been exposed to all kinds of different carcinogens. You know, like formaldehyde. They used to preserve all of our dissecting animals in formaldehyde. It's a carcinogen. They never knew that until, say, 10 years ago. I'm like, oh, wait. I never put on latex gloves doing dissections. Really? It's now a carcinogen? Y'all, for sure, you know that. Yeah? Yeah, we know. Okay. Well, thank you. All kinds of things. DDT, they used to go around spraying the DDT, and we'd chase the DDT truck on our bicycles. It turned out to be a carcinogen. It kills the flies. So yeah, you could have picnics outside and so forth. It's grand, but a lot of people my age, you know, God only knows. And you ask anyone about my age, they'll go, oh, yeah, we used to chase that truck. You betcha. I don't know if your parents are about my age, ask them. They'll go, oh, yeah, we always chased it. Okay, so the tumor is the mass of abnormally growing cells. So if it's a benign tumor, that means that when the cells divide, they stay together. They don't spread apart. If it's malignant, they spread apart. That's a malignancy, which means if they spread apart, they can spread to other areas of your body. So I had a friend that he was going to school at SMU and just fell out of his chair one day. They took him to the hospital, ran some tests, finally got him into he went in to get a brain scan. Anyway, and it turns out he had like 7 tumors in his brain, and it was pretty bad. Anyway. That cancer, and they did some pretty unique features with him. They now have this device, and I don't know how you can be a candidate for this, but it's a laser machine that they shoot laser in from several different directions at a certain intensity, and then when the laser points meet, then the intensity goes way up. And so they can map out exactly, through an MRI, exactly what that tumor looks like, and then they can go in there with the lasers, and then they can destroy the laser and not harm the other tissue. It's very cool. And not many people, he was the first person to ever have that done. And they did a documentary about that, which was pretty cool. Anyway, I don't remember the name of it, but I'm sure you could find it. You could Google it, find it. But so I don't know what that's about. Anybody know what the box is for down there in the middle of the circle? They're collecting grass for compost? Really? Well, I know you can. You can do that. Yeah, okay. Anyway, okay, so malignant tumors, yeah, they spread, and that is not a good thing. It's called metastasis, and this is what happens. So this is a little bit of breast cancer, and then you can see the cells are spreading, and they spread into all these little lymph nodes. The breast, as well as really all over your body, you've got lymph nodes, and these are special vessels into which things can enter which are not supposed to be in your body. And one of their major functions is to collect fluid that leaves your blood and is not recollected into your blood. 90% of the fluid that leaves your blood is mostly water and nutrients and that kind of thing, is recollected back into the bloodstream. But 10% of it stays out in your tissues. Well, if you always have this 10% going out of your blood vessels, staying out in your tissues, you would blow up and blow up and you'd blow up eventually. Blow up. But something has to reconnect all of that and put it back into your circulatory system. Well, that's what the lymphatic system is for. One of the main things. The other thing is they have lymphatic nodes that tend to that have macrophages, which are these cells that engulf things that shouldn't be in your blood, like cancer cells, as an example. And so that's one thing they do is they go in and they check the nodes, like the axillary nodes in the armpit. If you have breast cancer, they'll check your nodes out. And if they find any cancer cells in the lymph nodes there, they rip those puppies out. Your nodes are gone. They don't want, because it could spread to other parts of your body, and that's not what certainly anyone wants, but it does happen. So you don't have to live long. I think, what is it now, 1 in 7 women will get breast cancer? Is that the rate? It's 1 in 8 or 1 in 7, somewhere in there. I remember the day when it was 1 in 10. Now it's gone up. It keeps going up. Now it's like 1 in 7. It's like Angelina Jolie. Cut them off. Take them. If it runs in your family, that kind of deal. I can understand it. Absolutely. Brave woman. I'm not sure how Brad felt about that. That probably is a bad, ugly, nasty, bad joke. But anyway. Okay. Now they can do a lot of personalized treatment, which is really cool. They genetically sequence the cancer, and then they can build drugs to that particular genetic sequence. So it's getting better. Thank goodness. Hopefully someday they'll eradicate that. Now, this is the part that I think most people have more trouble with when you're talking about cell division. Meiosis. So this is reductional division. We're going to have to take cells that have 46 chromosomes, and of course those cells are in the gonads, which will either be the testes or the ovaries. I'll let y'all figure that part out. Testes or ovaries, and we have to reduce the chromosome number in half. So we have to go from 46 to 23, and it does take 2 cell divisions to do that. And then during the process of dividing the cell 2 times, there are different points at which you can create genetic variability. And so that means that the probability that any 2 sperm would be identical even from the same person is not very probable. The probability that any 2 eggs produced by any one female would not be very probable. So the probability that having the same sperm and the same egg unite at any 2 different times, not very probable. They call that random fertilization. Even if you have the same, even if you have the same partner producing a child, the probability you would ever produce 2 children that were exactly the same is like 1 in over 64 trillion chances. And that doesn't even include this thing called crossover right here. And when you include that, it's like off a chart. It's astronomical. The probability goes to almost nothing. It's not going to happen. So every child is going to be way different, way different. And I mean, you look at you and maybe a sibling, or if you have friends that have children, you look at their children and you go, man, those kids are all so different. And it's like that. All my friends' kids, it's like, no. Nothing similar about those 2 children at all. Have different interests and personalities, and they're different. They don't look the same. Yeah. The new building at Odessa College called Woods Math and Science today just blew down. in a 20-mile-an-hour wind. Yeah, it gets really much worse than that in Odessa, Texas, but in a 20-mile-an-hour wind, it just completely crumbled. Smushing all these people on the inside. That's horrible, horrible. Well, we shall bow our heads in a moment of silence. I hope not, but we got to go. Today's the day. I'm down. I'm in. Count me in. Anytime is a good day to go. They don't make a bad time. They're all good. Okay. Okay, so somatic cells, these are body cells, anything but a sex cell. But a sex cell, a sperm or an egg. All the others that you have are called somatic cells, which just means body cells. Somatic cells, body cells. not a sex cell. Okay, so that kind of clears that up. Homologous chromosomes, we may have beaten this thing to death at this point, but it doesn't hurt to repeat. You get one of these from your mother's, number one of these from your father, number one. So you get 2 ones from each, and one from each parent. And you get 2 twos, one from each parent, and 2 threes, one from each parent, and 2 fours, one from each parent, blah, blah, blah. Same length, same size, carry the same traits. A locus is where you would find a gene on a chromosome, but of course on the other homologous chromosome, sometimes referred to as its homologue, on the homologue of the first one, at the exact same position, you have a gene for the same trait. Now, the traits may be different. One may be a trait for brown eyes on one homologous chromosome, and on the other, it may be for blue eyes. But the trait is still eye color. So, locus just means location. Fancy word, location. Where's it located? Of course, we do get, most people, not everybody, most people get 1 pair of sex chromosomes. XY is male, XX is female. Some people get 3 XXY. That's called Klinefelter's. A person who has Klinefelter syndrome, they actually develop breasts because they get 2 X chromosomes, but they also develop penis and testes. The problem with that is the fact that the penis and testes never fully develop. So they never go into sexual maturity. So they never go through puberty, and it's like a 6-year-old. Sex organs of a 6-year-old. The breasts develop beautifully, though. Good breasts, just nothing else works. Anyway, it happens. And then you sometimes end up with a person that only gets 1 sex chromosome, like 1X, and that's it. They call it XO. That's called Turner syndrome. And Turner syndrome individual would be someone who would develop female because they did get an X chromosome, but they never fully mature. Sex organs never fully mature at all. So the sex organs are that of a 6-year-old, and of course they don't produce the hormones, so they're not going to ever become an adult female. You know, it just doesn't happen. And that's a Turner syndrome. I have a good friend, and her child has Turner syndrome. And beautiful child, absolutely beautiful little girl, beautiful little girl. But of course, at her age, which is prepubescent anyway, it's not in the least bit odd. Nothing is odd about it. She's like a little girl. But when she gets a little bit older, she's be going, wait a minute, you know, all these other people are like hooking up and I'm not. You know, I don't really know what would be going through her mind, but I'm sure lots of things would be going through her mind with that. Anyway, the autosomes are everything but the sex chromosomes. So the other 22 pair. You get 1 pair of sex chromosomes, and then you get 44 or 22 pair of autosomes. Have nothing to do with the sex of the individual. We already did that. We did that too. We did that too. All right. So here's a pair of chromosomes that have duplicated. So we have sister chromatids of a homologous pair. And you see that they're banded, and they really do band, and these tell you where the genes are located. So you can find them pretty easily. Not hard to find a gene. Of course, they've mapped out the entire human genome at this point, which is very cool. They've done that. Okay, so we're diploid. We get 2 number 1s, 2 number 2s, 2 number 3s, 2 number 4s. You get it at this point. Gametes, of course, are going to be haploid. Only one of the number 1s. It could either be the mothers or the fathers. And one number 2, it could either be the fathers or the mothers. Or it could be chromosome pair number 3. You're only going to get one of those. It's either the moms or the dads, or it's the dads or the moms. Okay. So, of course, fertilization, sperm meets egg, and you get a zygote. Y'all know how this works. I know your parents all sat you down and had that conversation. I mean, I know mine never did, but we're like, I guess you'll get it somewhere. Yeah, I figured it out. That and so many other things they never would have dreamed of, really. blew that wide open. Anyway, okay, so here we go. All right, so we have to go through meiosis to do that. And so this is basically just an overall generalization of what happens. You start with a pair of chromosomes, and we would have, of course, 23 pair. This is only one pair. The blue one, we'll say from dad, the red one from the mom. They have to replicate first, so they do. We would have actually, instead of just one pair replicated, you would have 23 replicated in a cell within your gonad. One cell within your gonad. Okay. And then meiosis, you pull apart, the first meiotic division, you pull apart homologous chromosomes. That happens in anaphase 1. And then in the second anaphase, anaphase 2, so you go through prophase 2, metaphase 2, anaphase 2. Anaphase 2, that's when you pull apart sister chromatids. That's why it takes two cell divisions to get there. So really your haploid state begins at the end of meiosis 1. Because here, you know, when you start with two chromosomes and you replicate those, and you have sister chromatids, two sisters for red and two sisters for blue, you have a diploid number of chromosomes. You've got two copies of each chromosome. And then you pull those apart. Now you only have one copy of each chromosome. So really it's already haploid at the end of meiosis 1. That's when it is haploid. But again, as long as your two sister chromatids are held together, that is considered to be one chromosome. So you would actually have 23. Some of them red, some of them blue in both cells. So they're haploid at that point. So, and then we restore genetic constitution by fertilization. So interphase, you have interphase just like you do in mitosis. And then we go, so that's when they replicate, chromosomes. Prophase 1, nuclear membrane breaks up. I'm not sure dissolve is a good explanation. And the chromosomes condense, tightly coil up. I guess that's another way to put it. And then this is where something happens that does not happen in mitosis. You get a synapsis. The one from your mommy and the one from a daddy pair up. How do they know? Hey, you, come over here. You're supposed to be with me. I'm chromosome pair number 3. Remember? Hello? I'm a 3. You're a 3. Come over here. Yeah, we'll pair up. We'll match up. And we get so close that they can exchange chromatid material like the one from the mom and the one from the dad. That's called non-sister chromatids cross, which is crossing over. Non-sister chromatids cross. That's what happens there. And that creates a lot of genetic variability. So you can see right, well, I guess it's on this chromosome. Right here. You get a crossover right here. Those are non-sister chromatids. A red one from the mom and a blue one from the dad. And that's a crossing over right there. Oh, there's a crossing over right there. And there's a crossing over right there. You know, if you've got a total of, you think about it, 23 pair, you can have all kinds of crossovers on each one. Man, you got a whole bunch of different variations created. Because normally this little piece of red is not supposed to be on, that looks weird. Anyway, this little piece of red is not supposed to be on that blue chromosome here, and that little blue piece is not supposed to be on mom's chromosome, and so forth. So yeah, I mean, you get new combinations that you wouldn't normally get, which is very interesting. And so that, again, like I keep trying to hound on it, and you know where I'm, it's just all genetic variability. You want a lot of variation out there. Like I say, without that, we wouldn't survive. There's no way. Human could not survive. Nothing could survive. Plant, animal, nothing would be able to make it. I mean, in the course of time, things are going to change. The environments change. And if you don't have some variability within the population to be able to meet that change, you know, you get extinction. And certainly there have been many, many extinctions over time. We don't want to be part of that. We want to continue as humans. And, you know, I like to be optimistic and think that we will continue. You know, we'll face adversities, but, you know, okay, or y'all will face adversities. Let me say that. I'll be dead. But I'll be trying to help you. I think those who have gone before us try to help us. I'm okay. I know it's probably not just normal, but that's what I think. So, rely on those who have passed, because I think they're in your court. All right. So, metaphase 1, they call it a tetrad. Really, tetrads line up because this is it. You've actually got 4. Oh, sorry about that. My bad. My bad right there. All right. So, but really, what you have there, you have 4 chromatids. 2 of them are red sisters and 2 of them are blue sisters, but that's 4, so they do call it a tetrad. That's where that comes from. Well, they give it to you right here, tetrad. Since you do have 4 chromatids, and then 4 for this one and 4 for this one, and so forth. That's where they get that terminology. Okay. This kind of just kind of makes sure that I'm not sliding past something that I should have talked about and didn't. But you know, it's just like there's just never enough time. Not in an 8-week semester, for sure. Okay, anyway, meiosis 1, anaphase 1, homologous pairs separate. Now, if you don't highlight that, if that isn't in your head, you're going to miss it on a lab test, you're going to miss it on a major exam, you're going to miss it on the final exam, you're going to miss it on the comprehensive makeup exam, which, by the way, and I haven't really talked much about that, but tomorrow after the lab test, we will have a comprehensive makeup exam. So if you missed a test, whether it's lecture or lab, you can take it then. Or if you just want to take it for grins, you're certainly welcome to take it. Only the lecture test. I don't allow people to take the lab test just for grins, but the lecture makeup test, you're more than welcome to take it. In fact, I would recommend that you take it because it could be very similar to the final. It's a pretty good review for the final, actually. I would recommend everybody take it. So, after your lab test, I don't know, tomorrow, you know, I may just go ahead and give the lab test and then we do the comprehensive instead of class time. I mean, y'all can read these. Most of you go, well, I can read the PowerPoint. Thank you. Thank you so much. But I think I can handle that. Prophase 2, metaphase 2, they line up. Anaphase 2, sister chromatids separate. Sister chromatids separate. That's anaphase 2. So that's when sister chromatids are separating right here. So first anaphase, the homologous chromosomes separate, and during the second anaphase, anaphase 2, sister chromatids separate. And that, of course, along with the fact that you get crossovers at prophase 1, and then also, depending on how they line up let's go back up here. I want to show you this just real quickly. How they line up. Well, where is it? Nope. Okay, this is it. At metaphase 1, notice on this, I've got dads and moms and moms and dads and dads and moms. It doesn't have to be. I could have dads and moms and dads and moms and dads and moms. Or I could have moms and dads and then dads and moms and then moms and dads. Depends on how they line up. And that gives you different possibilities for gametes. That's called law of independent assortment. Each pair assorts independently of the other pair. The first pair, one goes to one pole and the other one goes to the other pole, but you don't know which one's going to go to which pole. Blue one up, red one down, or red one up, blue one down. That holds true for every pair. Dad's up, mom's down, or mom's up, dad's down. That holds true for every pair. So you get over 8 million different genetic possibilities for any one gamete. So that's why we say any gamete you produce is one of over 8 million different possibilities. Same thing true for the other person. One of over 8 million different genetic possibilities for any one gametes, and that doesn't include crossovers. So if you combine those together, that offspring is 1 over 64 trillion possibilities. And then if you throw crossovers in that, you will never have two children the same. Just isn't going to happen. They'll be so different, and you'll be like, I don't know where these came from. You'll be blaming each other. No, he's your child. No, he's your child. You know, but it's called genetics. That's what it's called. You don't have to blame anybody but God. He did that to you. Yeah. Want to blame somebody. There's your blame. This is a karyotype, just a visual display. This shows incidence. I do want to just real quickly just show this real quick of having a child with Down syndrome per 1,000 births. Okay, notice at age 20, and then it begins to go up. Incidence goes up at 25, 30, 35. When you hit 40, you know, I don't think that's very wise. You know, you can freeze your eggs way back here at 20. Have some frozen. You know, pick out a daddy and say, hey, here's a cup. Here's a cup. We'll freeze those. We'll freeze my eggs. We'll freeze your sperm. You know, and if I still like you after 20 years, then hey, we may do something about that. I think it's a very cool idea myself. It's like, you know, you don't have to be saddled with a kid at this point in your life. You probably need to be worried about getting through school. Some of you have some really, really ambitious ambitions, and so, you know, I would hope you could carry those through. You know, get that done. So yeah, freeze those puppies now. Get them frozen. I vote freeze. Freeze gametes. Yeah. You know, pick 2 or 3, 5 guys, 10, 20 guys. You know, write a description over each one. Just have them donate. You know, and then decide who you're going to have a child with later. They don't even have to know about it. You could write them a letter, a little email, text them. Having your child today, just went into labor. Okay, anyway, you guys have yourself a beautiful day. You've got a test in the LRC library. And I will go get those about 5:30 when they close, and hopefully I'll get them graded by tomorrow.